video. Dr. Hudson here again. In our last video, we talked about excitation, contraction, coupling, and in the video before that, we talked about the neuromuscular junction. So far, we've actually seen a big chunk of how skeletal muscle contraction occurs in the human body, in most animals for that matter, where we first went into how an action potential from the brain is going to end up creating an end plate potential voltage along the motor end plate, which is going to create a new action potential on the sarcolemma, and then that action potential travels down to the T-tubule to cause the release of calcium from the sarcoplasmic reticulum. So the way I teach, redundancy is the key to learning, so I'm going to review that really fast. Y'all ready? I wouldn't recommend trying to take too many notes on this. Just listen and see if this makes sense. If it doesn't, go back and watch those old videos again and take notes on those, and then come back. Okay. First and foremost, action potential from the nervous system travels down the axon, and it activates voltage-gated calcium channels on the bouton, which allows for calcium influx into the terminal bouton. Calcium concentrations increasing inside of the bouton drives exocytosis of acetylcholine into the synaptic cleft, where that acetylcholine then binds to nicotinic receptors, which allows sodium to rush in faster than potassium can leave, creating a net positive charge on the inside of the motor end plate called an end plate potential. That end plate potential spreads out back to the sarcolemma, where it's going to create a threshold event to activate voltage-gated sodium channels. Sodium rushes into the cell, and we have ourselves an action potential. It gets shut off as potassium leaves the cell, also activated by that same voltage-gated, same system for the voltage-gated sodium channels with the voltage-gated potassium channels. We just get a repolarizing event. Now that action potential travels across the entire sarcolemma but dives down into the T-tubules where it activates dihydropyridine, DHP. The DHP is attached to the ryanodine receptor on the sarcoplasmic reticulum, and that coil tightens, opening a gate and allowing calcium to rush out into the ICF onto the sarcomere down below. And that's where the next part of the story comes into play, is now we need to look at the sarcomere. Since the sarcomere is the smallest functional unit of muscle, that's where the work is done, because what's the function of muscle? Contract and relax. To shorten and relax. And as it shortens, it creates force, and as it relaxes, it lessens that force. As it shortens, we can create movement. As it relaxes, we can lessen that movement. That all occurs at the sarcomere level. So let's draw a sarcomere really fast. A sarcomere is from Z-disc. Just start drawing some ziggy-zaggy lines, right? Z-disc. Z-disc to Z-disc. So we got a Z-disc here. And we have a Z-disc here. Those are the ends of the sarcomere. Extending from the sarcomere, growing on a protein called nebulin, is another protein called actin. The actin grows on the nebulin. So let's go ahead and keep our colors the same. I drew in a pre-picture here because I'm not very good at drawing. It took me a while to do that one. But growing, polymerizing, balling together from the Z-disc, we have actin. And that's going to extend out towards the middle of this whole mess. Okay, so these actins are extending out. We're not going to make this perfect because that's just too much work. And you don't need to make it perfect to study. You just need to have some representation that allows your brain to start to visualize the process. This is actin, and it's the start of our thin filaments, if you've ever heard of that. Now, I used black for myosin, so I'm going to keep that consistent. in between each one of these actins, not attached to the Z disc, but running in between each one of these, is a thick filament. And we're going to go ahead and thicken this thing up. Okay, we're going to just make it thicker. Again, I'm not a good artist. I don't pretend to be a good artist. I don't need to be a good artist to understand physiology, and neither do you. This is myosin. And myosin is physically attached to a band of protein that run down the middle of this called the M line. The M line, the middle line. M for middle, right? The M line is what kind of holds the myosin in place, along with some other things like tightening. The red again was actin. And the black here is myosin. Think I have enough room here? Woohoo, I do. Okay, actin and myosin. And those are the two major proteins. Is that true? Kinda. They're all important. Those are the two major proteins that people think about when they think about muscle contraction, but they're not it. They're not the only ones. But let's finish our information on myosin. Myosin has a whole bunch of myosin heads that extend out, and they come into contact with the actin on either side. Those are myosin heads, and they're all held in place by these myosin tails down below. So these lines that I drew here, in essence, when you zoom in, they look like this. Each myosin head has two oval extensions that protrude from it. And then all of the myosin tails twist together, and they create these big, thick myosin structures that run from M line in, or that attach to the M line. Okay, what else do we have going on here? We also have we have a protein that wraps itself around actin. Now, I'm not going to draw it on every single one of these because that'd be a pain in the patella and also distract you from what you need to know. But what we have here is a protein that wraps itself around myosin, and as it does, it actually covers these little blue dots that I drew in the picture down below. Those little blue dots on the actin are called myosin binding sites. So these are myosin binding sites. And the protein that I'm talking about, I like to call it Twizzler because it wraps around and looks a little bit like Twizzler. But this is called tropomyosin. And tropomyosin covers, to the best of my ability, is going to cover those myosin binding sites. And there are two of them that wrap around. Again, I don't pretend to be a good artist, but as long as those tropomyosin are covering the myosin binding sites, then the myosin heads down below, they don't have a place to bind to. So how are they supposed to bind to that? They can't. Let's change our color up here to keep our colors coordinated. So the green now is going to be tropomyosin, and I just draw in a single Twizzler around my actin there. So what is going to allow the tropomyosin to shift off the myosin binding sites? Well, that's where, what color have I not used? I'll just go back to this light brown. I like this. Actually, I got blue here. That's where troponin comes into play. And troponin is a protein that actually has 3 distinct subunits to it. I'm just going to draw it in as one unit. And troponin is on each of the tropomyosins, basically. It's covering the tropomyosin, this big protein that hangs off of it. You guys can go online and look at pictures to get a better idea of this. On my drawing up here, I'm just going to draw troponin on each portion of the tropomyosin wave. An easy way to try to remember troponin is I like to think of Carlton from Fresh Prince of Bel-Air, and when he used to go, jump on it. Dun, 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 right? Troponin, you can go in the same rhythm, and it helps you remember troponin forevermore, because troponin is really important for this entire process. Troponin is the calcium binding site that's going to activate muscle contraction. Write that down. Troponin is the calcium binding site. This guy right here is a really important regulatory protein. It helps regulate this entire thing. Because when troponin gets calcium bound to it, what it does is it takes the tropomyosin and it allows the tropomyosin to shift off of the myosin binding site, which is going to expose those myosin binding sites to the myosin head down below. So that's one step of the crossbridge cycle. That's going to begin the process of crossbridge cycling. You must have calcium in the system, and calcium must activate troponin. Therefore, troponin will physically change the conformation of tropomyosin to shift that tropomyosin off of the myosin binding sites. We're not done yet, because now we have to think about myosin, okay? And there's no real good way to do that. It's not true. There's a lot of great animations online. I highly recommend that you go look at crossbridge cycling online and look at animations and videos, because they give you the best picture in your mind. They're the real deal, right? What I can try to do is explain how myosin heads go through 4 major steps, and there are a few more steps in there, but it's easier to start to learn them as 4 and then start adding in the minutia. Okay, the first thing down here with this myosin is that we have 2 states. We have a low energy state. This is a low energy, how do I spell energy? There we go. This is a low energy myosin, and this is going to be a high energy myosin. So what makes it a low energy and high energy is all related to the energy of the human body. And what is the major energy of the human body? ATP. Right? Myosin is actually also known as another name called myosin ATPase, which means that the myosin head is an enzyme that helps to hydrolyze ATP into ADP and inorganic phosphate. So myosin ATPase is going to hydrolyze ATP into the subsets of its parts. It's going to release energy, in other words, ADP plus inorganic phosphate, which is P lowercase I with the circle around it. That's our inorganic phosphate right there. Okay, that's the myosin ATPase. So in a system where there's not ATP bound to the myosin head, then it's low energy. But ATP has a super high affinity for this myosin ATPase. ATP is constantly trying to search out this myosin head. So here on the myosin head, this purple dot that I'm showing right here, this is an ATP binding site. I'm not sure if y'all can read that. I hope you can. Zoom in if you can't. It's an ATP binding site right there. And when ATP binds to that site, ATP gets hydrolyzed into ADP and inorganic phosphate. And ADP and inorganic phosphate are holding on to that same site. They stay there. And that energy that gets released allows this myosin head to go from this position to this position. And this is called the cocked state. Okay, this myosin head goes into the cocked state, and the ADP and inorganic phosphate are still bound here. So we have ADP and we have inorganic phosphate bound to that binding site. By the way, this light blue dot that I'm showing right there, that's the actin binding site. So that is where the myosin binding site on actin looks for the actin binding site on myosin. Hopefully you guys get the point there. So ATP just bound to the ATP binding site. It becomes hydrolyzed into ADP and inorganic phosphate, and that cocks that myosin head. As long as the myosin binding sites are exposed on the actin, then the actin binding site, now that it has energy, the actin binding site binds to the myosin binding site. At which time, you're going to just have to write this down because I'm running out of room on my board. At which time, the first thing to happen is inorganic phosphate falls off. And when inorganic phosphate falls off, it causes the myosin head to bind even tighter onto the actin above. Alright, so imagine this is actin right here, this is a myosin head. I go into the Cox state, and as long as the actin and myosin binding sites recognize each other, then the myosin head goes up and binds to that site. Then the inorganic phosphate falls off, and that myosin head grabs on even tighter. And then the ADP falls off, and that causes the myosin head to go through a power stroke. And when it pulls, because it's bound to the actin up above, when that myosin head goes through the power stroke and pulls, it yanks that actin along with it. And that is crossbridge cycling. This is not the perfect video, but I sure hope that it helps you guys start to visualize this. And then you can start to write it down in your own words. Okay? The way that this works is it's a cycle. 1, you must have ATP bind to the myosin head. Now when it does, ATP gets hydrolyzed into ADP and inorganic phosphate by the myosin head. That allows the myosin head to go into the Cox state. Okay? That sets it into the Cox state. The high energy myosin. As long as the myosin binding sites are exposed, we must make sure that we take that note into play. As long as the myosin binding sites are exposed, then the myosin head binds to the myosin binding site on actin. What happens next? I'm going to draw a little arrow now. First, the inorganic phosphate falls off. And when that happens, the myosin head binds tighter. I'm just going to write in tighter because that's the room that I have. You guys have to do the rest on your side. When the inorganic phosphate falls off, the myosin head binds tighter. Then the ADP falls off. And that allows for the power stroke. And since the myosin head is bound to the actin starts to slide along the length of the sarcomere. 3, we're actually going to end up cycling right back around again. And these steps, you can make these steps in any way that you want, right? It could be ATP gets hydrolyzed. Therefore, we're in the Cox state. That could be step 1, right? Or we're in the Cox state, myosin head binds to actin, that's step 3. You can make these steps any way you want. Studying's great like that. Okay, when the ADP falls off, we have the power stroke. Then what? Well, as long as ATP has not been hydrolyzed by the myosin head, then the myosin head stays bound to the actin. Okay? If there's no ATP, what we do is we call this the rigor state. Because remember, the only time in your life that you don't have ATP is when you're dead. And that is rigor mortis, when all the muscles are contracting and then the myosin head stays bound to the actin. That's what causes rigor mortis. If no ATP, then rigor state, but if there is ATP, what do? We go back through the cycle, the Crossbridge cycle. Okay, well, I certainly hope that that was helpful, that it leads you down the right track of studying, and best of luck in your endeavors. Watch this video again if you need to, and ask any questions that you need to as well. Humankind, be bold. See you in the next video.